Hello and welcome to our webinar Wednesday lineup. I am Helmut Prechel, Product Manager for Molecular Biology at Biosym. We are proud that so many people, customers, and curious scientific workers join our webinar today. Thank you very much. The aim of Biosym webinars is to transport new and exciting information about laboratory technologies and new products to all interested people. Today, we would like to present a quite new instrument which enables cell counting without slides, the CellDrop Automated Cell Counter. And we are pleased that Andrew Jones from Denovix will introduce the exciting device and technology. Andrew is Market Development Manager at Denovix, which much more than a decade of experience in microlighter spectroscopy and cell counting technology. At the end of the session, we will have a few minutes for discussion. Please type your questions via chat into the system whenever you like. We will try to answer them at the end. Thank you in advance for your input. Andrew, now please give us an insight into CellDrop.
Thank you, Helmut. Thank you, and thanks for inviting me along today, and thanks everyone for joining us. So as Helmut said, I'm the Market Development Manager with Denovix. Denovix is a US-based company. It was established in 2012, and previously best known for our DS11 series of microvolume spectrophotometers. But last year we launched the CellDrop Automated Cell Counter into the market, and it's been really well received. And what I'd like to do today is to give you an introduction to the CellDrop. I'll take you through a few slides, not too many, and then we'll switch pretty quickly to basically a live demo. So I'll show you how we run samples and some of the key features around the CellDrop that makes it so different to any other cell counters on the market. So some of the key features, as Helmut just mentioned briefly, one of the key things for the CellDrop, key values in the market really, is that we enable you to count cells without slides. And this is based on the unique technology that Denovix has developed called DirectPipette Technology. And I'm going to take you through that in a little bit more detail and show you how that works in the next slide. But some of the other key features that we'll see during the demo is that we have a dual fluorescence and brightfield optics system. And we can choose between dual fluorescence, brightfield, or just brightfield only. So we can run cell counts and accurate viability studies. The instrument is networked or networkable, so we can connect via the Wi-Fi or through Ethernet. And we can export our data through email, network and label printers. We can save to USBs, and all of our data gets saved on board. We can save many thousands of samples on the inbuilt memory. And we'll see that the CellDrop is powered by a 7-inch HD touchscreen, so we can get lots of detail on our cells and get a very high resolution image of what we're looking at. And I'll take you through the software and how that looks as we go through the presentation. So DirectPipette Technology, as I mentioned, this is the core technology behind CellDrop. And it fundamentally consists of two parallel permanent wipe clean sapphire surfaces. Okay, so you can almost think of these as like two sapphire slides facing each other. And the surfaces are separated and the distance set by a high precision screw. And I'll show you that as we go through the demo. And basically what this allows us to do is to set a chamber between those two surfaces that is analogous to your hemocytometer or to a disposable plastic slide. So the cells are pipetted directly into this chamber, but it never goes into the instrument at all. It's held there by surface tension in a similar way to a microvolume spectrophotometer like the DS11 series, or if you're familiar with the nanodrop type system. So surface tension holds our sample in place. As we pipette our sample in, the cells will drop to the bottom surface, and that's the surface that we image. Once we've imaged the cells, all we then do is wipe the surface clean, and we're ready for our next sample. So some of the benefits of this direct pipette technology, clearly we're not having to use any disposable slides. So there's an environmental aspect to this, that we're using less plastic in our labs, which is more and more an increasingly important aspect of what we're doing, is to try and minimize the environmental impact of our research. Clearly, if we're not purchasing consumable slides, we're saving money on those purchases. So we're not having to purchase the slides, but also we don't have to pay to dispose of those slides. We don't have to pay to dispose of hazardous waste. I mentioned earlier that we're able to control the height of the chamber. So your standard hemocytometer is likely to be 100 microns deep. And that's the same with disposable plastic slides generally. And that's our standard setting, so it's very familiar and very similar to your hemocytometers. But what we're also able to do is, if we have a very high density cell culture, we can actually set a lower chamber height and so pipette less volume onto the measurement surface and so fewer cells. And conversely, if we have a very low density cell, we can actually make a higher chamber and pipette more samples so we can get a more accurate measurement. And what this allows us to do is have the greatest density range of any instrument in this sort of class, and also the largest cell size range. Really, having a greater height allows us to measure bigger cells, so you don't have those issues with plastic slides where cells get caught along the inlet groove, especially if they're too big. So the cells flow easily into this chamber. The software we're going to go through on the live demo, but it's important to note that the software is really designed by life scientists. So we have life scientists sitting next to our programmers and our IT department that work in concert together to develop this software. So what we like to think of, and the feedback that we get, is it's a very intuitive piece of software, very easy to use, and has all of the key features that you want in a cell counter. It's fast, so we'll see that brightfield measurements only take about 3 seconds, and we can measure all 3 channels in around 8.5 seconds. We can set up user accounts that can be password protected. We have custom applications, custom protocols. So if we don't have an application that's specifically for you, there are lots of options to customize this platform. And as I mentioned, we can store thousands of samples on board, and we can export the results in a variety of formats. Now, the team at Biozine will be able to take you through all of the options available for CellDrop, but broadly there are 2 different models. So we have a brightfield only model that is generally used if you're just looking at very clean tissue culture samples and looking at maybe trypan viability. That's the brightfield only. If you're looking at things like primary cells, if you have debris in your samples, if you're interested in really improving the accuracy of your viability and cell counts, or if you're looking at GFP or yeast measurements, then it's likely you'll be interested in the fluorescence system. This is dual fluorescence, and it's red and green fluorescence. Alongside the instrument, we do have a range of assays that are tailored for the instrument. Now, it is an open platform, so if you currently use Trypan Blue in your lab, you can continue to use that, or you can purchase the Denovix optimized assays, and likewise for our fluorescence assays as well. We also have some fluorescent beads for brightfield and fluorescence, and those are essentially just to check the performance of the instrument, or to demonstrate the instrument, and those are what I'm going to use today. So I'm still working from my my home office, So we don't have any cells available. But I'm going to show you the characteristics of the instrument using some beads. Okay, so I'm going to switch over to sharing my screen for a live demo now. And so you should be able to see the CellDrop. And as mentioned, the instrument that I have here today is the CellDrop FL. So this is the fluorescence, the dual fluorescence instrument. A little guided tour to the instrument. That as I mentioned, the software is all controlled by this large high definition screen. We have 3 USB ports to the rear, and we have an Ethernet and an HDMI output. So if you want, you can output the screen, what's seen on the screen, to a larger monitor, and you can view it on a large monitor. We also have an additional USB port to the side of the instrument here, and this is generally used for data export. To the top of the instrument, you can see that we have our measurement arm. And the measurement arm, in a very similar way to microvolume spectrophotometers, we can lift that up. And you'll see on the bottom is where we have our sapphire surface. And there's a second surface on the upper arm itself. And just in the middle of the key plate on the top is our high precision screw that sets the height of the top arm and so the depth of our chamber. Now, we pipette our sample in with the arm closed rather than the arm up. So I'm just going to zoom out a little bit, and you might see, if I just come in slightly briefly, you might see on the left and on the right hand side of the measurement surface here, you have little grooves. And the grooves are there just for alignment. We don't pipette our samples into those. It just makes it super easy to load our sample. And I'll show you how this works. So looking down towards the software here. There we go. So looking down towards the software, all of our applications are separated into different measurement apps. And we have brightfield, trypan blue, AOPI. I'm going to select trypan blue to start, so we can take a look at this. And I'm just going to load my samples in. So I'm loading 10 microliters of sample because we're using our standard height of 100 microns. And so all we do is we just slide the tip up to the arm, pipette our samples, and you'll see all of the cells rushing in. So this is a live image that we're seeing of that measurement surface. I'll just move it down slightly. So you can see that all the cells or the beads have now settled on the bottom and they've stopped moving. And in this particular sample, these larger beads with the white centers are supposed to represent live cells as we would see them under the microscope with trypan blue. And there's some smaller dark cells, dark beads that are representing our dead cells. So those are the ones that would have taken up the trypan blue stain. So we've loaded our sample. If we want, we can define protocols. So we could add a protocol, we can use a predefined protocol, or for the purposes of this, we're just going to use our default protocol to run these samples. We can add a sample name. So if we want, we can put barcode readers onto the system, a USB barcode reader, or we can just put in manually our sample name, a description, and a passage number if we want to as well. All we're going to do is we've got the default protocol here. I'm just going to hit count. And it's going to go through taking the image and looking for any of those bright centered cells And it's going to take the bright centered cells and say that they're live. And any of the dark cells, it's going to identify as dead. So hopefully you can kind of see this. All of the live cells have been surrounded by a green circle, and all the dead cells have been surrounded by red circles. Now, if we find that anything has been measured that we don't want measured, or if we want to optimize the settings slightly, we can optimize it after we've run. So this is a great way of starting with a default protocol, and if we want to optimize it for our particular cell line, we can come in and we can play and manipulate all of the settings available without having to run multiple samples. So some of the options available, we can specify a size range for our cells. We can tell it how round our cells are, how complete the cells are. We can set thresholds for how dark it is before it's counted dead. And we have some options here around counting very small cells or counting irregular cells. So we can change any of these protocols, or sorry, any protocol settings. And we can, so for example, maybe we wanted to set a different diameter range, and I wanted to set that to 20 rather than 30. And then all we do is hit count with current settings, and it will go through and do that reanalysis for us based on the settings that we've supplied. So again, this is a nice way to optimize for your cell lines, because then if this is a better set of parameters for you, you just save it to a protocol, and there's your new protocol the next time you run this cell line. A couple of other options. We have the option to gate based on size. So we can get this histogram. And if we know that our cells are in a particular range, we can just exclude anything that's outside of that range. So if we have some debris that's being picked up as cells, we could just exclude those based on cells, and again apply that to the current sample. And on the left-hand side here, you'll see that all of our sample information is reported and updated as soon as we change any of our settings. So we have total cell count, a live and dead cell count, viability, diameter, total cells per mil, live and dead cells per mil. When we're happy with our sample, all we do is we take a normal laboratory wipe, lift the arm, and wipe our sample off. So we just wipe the top and the bottom measurement surface. And then we hit next count, and we're ready to measure our next sample. So I'll pop one more sample in. So you can see the tsunami of cells. And we'll hit count. So as soon as we lift the arm to take the next measurement, that saves all of our data to the onboard hard drive. And once we've got a couple of samples in there, we can start to view samples next to each other. So we can view it in a table format, where we just get the live cell count, the diameter, the date, the time. Or we can look at the images or overlay the samples on graphs. And there's a whole range of different options for different graphical views that we have. So for example, live and dead cells by size. If we're interested in cluster sizes, we can take a look at that. Or just live cells. So both of those samples next to each other, and we can do a whole range of samples in our batch like this. As I mentioned, the exporting options are really powerful for the CellDrop. we have a few different options about the format that we export. So we can export it in a .csv. So that's just a comma-separated format that you can open up in Excel or is compatible with LIMS systems, those sort of things. And that will include our results and all of our protocol settings. Or we can generate a PDF. And in the PDF, we can define results and protocol settings. We can ask for images. And also, we can export those histograms that we were looking at. And the third option is that we can actually export the raw images as well. So we'll say PDF for now. And we click Next. And it asks us how we want to export that PDF. The option is we can use USB. So we can save it to a USB. Or one of the most popular formats for exporting is to email. So we can set the CellDrop up with its own email address, and we can save all of the email addresses of your lab or the people that use the instrument into the software. And so whenever you collect your data, all you'll do is select your email address, hit Next, and it will email that PDF report straight to your computer. So you don't have to worry about USB drives or saving the data or writing the results down. It's all going to be there waiting for you on your computer. And to give you an idea of the sort of format, this is the PDF that gets printed out or that gets sent. So you get all of your sample information, the images, and the histograms. The third option here is you can go direct to a network printer. So printing out that PDF, but it will basically go and look to your network that you're on and print it out directly to any network printer that you specify. We won't print that out for now. We'll go back to the count screen. Okay, so in this sample, I mentioned that there are different populations of cells. So there are dark, small ones, bigger, bright ones, bright centered ones. And there's also a lot of debris in here. So we deliberately made a sample that looks kind of challenging to measure. And you saw with our Trypan Blue, it does a great job of picking these out. But there are some subjective calls with Trypan Blue. So Trypan Blue can sometimes struggle to differentiate debris from cells, and you have to make a call. You have to decide whether it's a cell or whether it's debris, and you do that when you're manually counting or if you're automatically counting as well. And likewise, it's how dark does it have to be before you call it a dead cell? So one of the ways to get around this, if you're interested in improving the accuracy of your sample analysis, or if you're dealing with primary cells where you may have a lot of background cells compared to the cells of interest. For example, you might want to measure blood samples. And with blood samples, you might be interested in the white blood cells, but there's a big background of red blood cells. And for these, we recommend fluorescence. So I'm going to take you into one of the fluorescence apps, and we'll go for AOPI. So this is acridine orange and propidium iodide. Acridine orange will specifically only stain acridine orange will stain all of your cells in the sample, but they have to be nuclear dead cells. So it gets excluded from any live cells. So we can look at the two channels, the green and the red, and we can give a specific measurement for live and dead cells without the subjectivity that is associated with Trypan Blue. So if we come in, we just have a look at the sample. It's difficult to tell kind of which of these black ones would be live, which would be dead, but we have a channel selector here that allows us to look at the fluorescence, and you can see As soon as you start using fluorescence, it becomes very clear where your cells are. So your live green cells, and if we select red, it's a little more tricky to see on the screen, but it's very clear when you actually see the screen physically here. You can see that we have all the dead cells highlighted here. So we've got this sample on. All we do is we hit count. And what this is going to do is it's going to look for any green cells and call those live. It's going to look for any red cells and call those dead. And if we can zoom in on the screen here, and maybe I can zoom in on this a little bit. There we go. So it becomes really clear using fluorescence what is live, what is dead, and what is nucleate and what is anucleate. You can see, perhaps you can make out here that there are two cells that look the same size. One is clearly live and nucleate, the other is anucleate because it hasn't been stained. So it's a very, very powerful, very robust way to measure our cell number and specifically our live and dead cells. One other nice feature that I really like about the software is the dilution calculator. So if we've got our specific measurement of live and dead cells, but we want to adjust our initial sample to a particular concentration for our downstream application, we have a dilution calculator. So we can specify what target cells per mil we want, what the target volume is, and what our original volume was. We click OK, and then beneath our results here, we get our dilution guidance. So it will tell us that we need to dilute a certain amount of our sample into a volume of buffer. And so that just makes life a little bit easier for us. We don't have to do the calculations ourselves. And that data can also be viewed in the results. So it will show us our dilution guidance underneath. And that can also be exported. So if we wanted to print that off and take it with us back to the hood, we can do that there. Okay, so just going to get out of time. Okay, for time at the moment. I'm just going to show you through some of the other apps briefly. We obviously we can't go into too much detail in this short time that we have today, but we can always arrange additional demos for you. There's more information online. So we have AOPI and the option to do those two apps separately. One thing that a lot of labs are really interested in is the ability to run GFP samples. So if you're doing transfection studies, you can work out the efficiency of your transfection using the CellDrop. In a similar way to our previous apps, we just load the sample, hit count, and it will go through. And in this particular application, it's only looking at the green channel because we're looking at GFP. And you'll see it's pulled out everything that is fluorescing with GFP. And on the left-hand side, we now get an additional option where we can gate based on fluorescence. So in this particular application, what we're doing is we're saying that we want to eliminate anything from the background and to set a threshold over which we're going to call our GFP measurement successful, our GFP transfection successful. So you can see here, there's a big background of essentially non-fluorescent material, which is all those big black beads. And we can set the threshold just above it, apply that gating, and it will give us an accurate measurement of how many total cells there are, how many cells have GFP, and the efficiency of your transfection. So this is a nice additional application for our cell counter. We also have the option to measure yeast. So yeast is an application that I would strongly recommend using fluorescence for. Generally, we use fluorescein diacetate in our assays for measuring the yeast viability. And a lot of people are using the CellDrop for yeast now because our lower range of detection is so much better than sort of competitive instruments in this market. And that's a consequence of the DirectPipette technology and the high precision optics that we have. We have Trypan Blue and AOPI apps for primary cells. And they're similar to our standard apps, but the default values are really tailored towards the primary cells, tailored towards primary cells. So you have default values in there that are specific to get you started on a primary cell sample. We have custom methods. So if we don't have a method that matches your requirements, we can set up custom methods, and we can set up user accounts and passwords too. One of the final apps I'm going to show you really is the data app. So as I mentioned, all of the data that is taken or captured on the CellDrop instrument is saved to an onboard hard drive. And this is really important for labs that are working in a kind of regulated environment, but it's useful for all labs to be able to refer back to see how their cell cultures or their treatments progress over time, and to have an archive of their data. So this data app is our window into our database. Okay, so this allows us to search our database and all of the data that's been saved. And we can do that based on the applications. So we can use search criteria to find our specific cells of interest. So we can select from any of our applications, AOPI in this example. We can choose particular protocols. So if we've saved a protocol for a particular cell line, for example, we could search specifically on that, and we can tailor the date range as well. And you'll see that it generates, when we hit search, it takes less than a second to go and search all of the data on the instrument and report all of that back to us. So it'll come up with our sample name, the date it was captured, and the protocol. And we can go through any of these and just highlight them and add it to the report. And now all of those samples will be lined up in a report next to each other. So you can see the cell counts, the diameter, the date it was collected. And you could highlight those, highlight the ones that you want, and export those in a similar way to we saw with the measurement apps, the count apps. So you can generate reports out of any of the data that you've captured previously on the instrument. And likewise, if you want to go and view that data on top of each other in graphs, you can see the viability here, but we have access to all of that data that we saw in the count apps, and we can see all of those samples overlaid on top of each other, and we can select from the list and exclude certain ones and add others here. A few other kind of apps along the bottom here that are worth talking about. We have an onboard timer, an onboard calculator for things like dilution calculations. Onboard help, so if the user guide is all ready to go on board. And we have a software updater. So if you're connecting it to the internet, it will alert you to new versions of the software. And it's worth pointing out that we do constantly develop our software and include all our we listen to all our user feedback and try to include as many features as we can that are requested by our customers. So all of those software updates are free of charge. and they become available through the updater. If you don't network it, that's fine. You can do it by USB. So that's the kind of summary to the CellDrop instrument. It's a fast, accurate, automated cell counter available in dual fluorescence or brightfield. And a key feature of, or unique feature of the CellDrop is the ability to measure and count your cells without slides. So you can avoid having to buy disposable slides for the lifetime of the instrument, and avoid the environmental impact and the cost of that. So that's the end of my presentation. I think there may be some questions that have come through. I think Helmut has some questions there. So if you'd like to fire any of those at me.
Okay, Andrew, thank you very much for this nice and interesting presentation. So we have learned the CellDrop really eliminates the need for slides and enables a very smart workflow. If you're interested in Germany and Austria, you can contact Bison for further information or for quotes. And if you are interested in working with CellDrop in your lab, just contact us directly or our local sales account managers for a demo. And now we are open for questions. You can type them into the chat box and we will answer. And we will start with, what if my samples are hazardous? Is it safe to use them on CellDrop?
Yes. So, echo here. So yes, it's a good question, and there are two answers to it. So hopefully you can still see my screen okay. The footprint of the CellDrop itself is, if I give you an idea, this is an A4 piece of paper. So it's about the footprint of an A4 piece of paper. So it can fit within a flow hood, and it has a very narrow footprint to it. So that it's the depth of a flow hood and it can fit within a flow hood. But often what people will do is, especially in a kind of multi-user environment, they will have it sitting outside of a flow hood. But if customers have, or if users have particularly hazardous samples that they want to keep contained and they're worried about not having a slide, we have the option to also use a disposable slide. Okay, so if we look at the top here, this is where we would normally pipette our sample, but around our measurement surface, you'll see a kind of cutout, a slide-shaped cutout. So if particular people have, or if particular samples need to be contained, you can just use disposable slides for those. And we just set it in the protocol. We set it to slide rather than direct pipette. It takes 2 seconds to do. And so you can use it for, yeah. So if you have hazardous samples, you can either use a disposable slide, or this can be situated in a flow hood. If you do use direct pipette for it, you can use bleach to clean it if you like to, or ethanol can be pipetted into the chamber, and the surface itself, sapphire, are resistant to all of those sort of cleaning chemicals. Hopefully that answers it okay.
Mm-hmm. Thank you. Next one, what maintenance is required? Does the CellDrop need calibrating?
Yes. So the only real maintenance that is required is to keep the surfaces cleaned in the same way as if you're used to using microvolume spectrophotometers, similar sort of thing. So just wipe them clean, and you can, as I mentioned in the last question, you can pipette 70% ethanol in there to do that. In terms of general maintenance, that's it. In terms of calibration, every time you turn the instrument on, it will go through an initialization routine. and that sets the calibration. So it's so basically you don't need to check the calibration. It doesn't need a service engineer to come in and check it. It will always set that calibration every time it's started up.
Mm-hmm. Very good. Next one is, do you have recommended settings for different cells?
Yes. Okay, so that's another good one. So generally when we start off in a lab, the default protocols are really good for most cells. But there are modifications that people make, and there are cell lines that we've tested, and we have a list of kind of cell lines that we've tested and validated the protocol settings for online. So you can go, I mean, they're not password protected or anything, they're all there, and we add to them daily almost to the list of cells that we've used. So yeah, so we have kind of standard protocols that we recommend. Now, as pretty much everyone who does cell culture knows, cells in different labs perform in different ways. So it's important to have those starting places, but it's also important just to have very simple ways to optimize yourself as well. And hopefully you saw in the software, it's very simple just to change a few settings to optimize and then save or refine that protocol to your own cells.
Mm-hmm. Very good. So thank you very much again, Andrew. Our time is over, unfortunately. Other questions raised in the meantime will be answered by email. So you get an answer, definitely. Yeah. Thank you very much for attending the webinar. Have a nice day and goodbye.